Bioinformatics.
Okay, so welcome to another lab follow-along. In this one, we'll be going through a brief tutorial on how to assemble a genome. Again, I don't expect you to watch this start to finish. This is meant to be a resource that you kind of pick and choose pieces where you want to see how I did it, although you're more than welcome to watch all the way through. In this one, we are going to be assembling a bacterial genome and then eventually annotating it after playing around with some different assembly settings and seeing what can affect both positively and negatively the quality of your genome assembly. If you don't remember some of these terms, like what a contig are, or things like that, or a scaffold, go back and watch the lecture for this section as well. So there is some terminology here that came from the lecture. So first things first, we got to sign into our server, which I've already done, and we need to go ahead and install a couple things. So typically I have used conda install one by one for each package, but what I didn't tell you, because I like to start things off simply, is that when you use conda install, you can actually pass a whole sequence of package or program names and install them all at once. And so here we can say conda install -c from bioconda as usual. We can install Velvet, sickle trim, BWA, SAM tools, which you probably already have, but we'll put it there just in case you're on a new server, data mash, and seqkit, which you also might have, and we'll automatically say yes to all of them. Once all your applications or programs are installed, let's go ahead and get set up for this lab. So as usual, I like to make a lab directory that just reflects which lab we're on. You're welcome again to deviate from this. So we'll cd into the directory name, and because we're going to be doing some building of genomes and then also some mapping, I like to again start to show you a little bit better examples of how to be organized in your data structure. So we'll also make a subdirectory within the Lab 12 directory called refs, and we'll make one called data where we'll put the raw data as well. And then what we'll go ahead and do is we will download the Staphylococcus reference genome, and we will set that as an environmental variable. Now that was kind of a long line. What I want to point out is that we actually redirected it into the reference directory and named it s-aureus ref.fa. Okay, and so what we'll do is we'll go ahead and set this as an environmental variable, passing the full path of where it's actually located, so we don't have to constantly retype it wherever we're at. Okay, so we've got our ref there. Now, thinking about kind of more the experimental side of generating sequence data, if you know you want to assemble a genome and you have a rough idea of the organism's size, you can try and inform how much sequencing you should perform on your organism in order to build a decent quality genome. Now, this is a ballpark estimate because repeat complexity, type of organism, whether it's eukaryote, prokaryote, all have big impacts on how complicated the genome is. For the case of doing a bacterial genome and doing a mediocre job, 45x coverage is not a bad goal to shoot for. And so how do we calculate that? And so the basic equation is that coverage equals the number of reads times the length of those reads divided by the genome size. So the Staph aureus genome is about 2.9 million base pairs, or 2.9 megabases. And our reads from the experiment that I'm going to have you guys pull from are 101 base pairs. Now, in order to hit 45x coverage, if you went ahead and did that calculation, you would see that you need about 1.2 million reads. But these are actually paired end reads, so for every read we actually get twice as much information. We get 2 times 101, so we need half as many reads, 600,000 total reads, because they come as a pair. But just to be safe, we'll grab an extra 200,000. So what we're really going to shoot for are about 800,000 pairs. Now this isn't that many. I mean, typically you can get millions and millions and millions of pairs from a single run. So what we're going to do is FastQ dump this and actually subset it. Now, what we've done in the past is use FastQ dump and then subset the amount of reads we want. This is not really ideal because it subsets based in the order that the reads came off of the sequencer. So you just get the first 800,000 reads, which is not really a true random sampling, and you can actually tend to see that the first reads off tend to be lower quality. So while it's less than ideal, we're going to subset 2 million reads and then subsample from that. In a real situation, if you're not worried about download time, you wouldn't subset at all. You would download the whole sequence data set and then subset it. How we're going to subset it is using this seqkit sample function. Again, how do I know that that's there? One, I've used it before in the past. And two, you could go ahead and look at the manual for seqkit. We are going to subsample 800,000 reads from the _1 and 800,000 reads from the _2 data sets. And so let's go ahead and get these data. So we'll fastq dump 2 million reads. Okay, so now that the raw data are downloaded, let's go ahead and subset it using seqkit sample. So seqkit sample -2-n, we want 800,000 from the forward reads, and we will call those we'll send those to the data folder, and we'll call them F1, okay, just for simplicity's sake. And then we'll do the same thing. You can hit up if you want. Just be careful to make sure that you change both the output name and the input file name, and hit enter. And now we've got both of them. So now we're going to take these reads, and in the past we've actually mapped reads to an already assembled genome. But what if you're working in an organism where you don't have a genome yet, a non-model organism, for example? Well, this is where you have to take all your raw reads and do the best that you can to try and fit them together to make a reference on your own. And this could be DNA, this could be RNA if you're just trying to reassemble different transcripts, could be lots of different things. So we talked in lecture about what a De Bruijn graph is, and I'll mention that the assembler discussed below and used below, Velvet, is a De Bruijn graph-based assembler. So you can read more information about it if you'd like, but without getting too into the weeds, as I try to avoid doing when possible, we're going to do this as a two-step process. The first step is to use Velvet H, which stands for hash, to hash out all of our raw data into k-mers of a size that we ask it to. So here I'm telling it I want to make 31 mers or 31 base pair length k-mers. And here are the fastq input data. Here I'm telling it it's fastq data as well. Then what we're going to do is we're going to take those k-mers that were saved as this output name, Velvet single N. We're going to treat these as single N data to begin with because we don't know what the insert size is, and we're going to experimentally determine that. And so we can do Velvet G for Genome Assembly, basically, using this k-mer input. And it's going to then build our assembly. And now I'm going to tell you before we do this that the first assembly is not going to be that great because we're using paired-in data, which usually help us build a better genome because remember they kind of are a fixed distance apart. And when you do your initial sequencing, you know what the distance is when you do the library prep. But what if you're pulling down data and someone didn't document things very well? Well, we can use our initial genome assembly to kind of estimate the insert size, and I'm going to show you how to do that. So let's go ahead and run these different parts. We're going to say Velvet H, and we're going to call the output Velvet single end 31 base pair k-mers. We're going to do a short with fastq, and again, you can go and read what all these things are if this is a piece of software that you really are dying to know more about. And we'll use data f2.fq, and we'll hit enter. And this will take a minute to hash everything out. Once you're done hashing out all of the sequences, we're going to go and use Velvet G with that output and build a rough assembly. And again, this is a small genome, so this shouldn't take terribly long. All right, so let's take a look at the output. So if we go into our folder, Velvet SE and LS, we've got a lot of different things. Some of these were our hashes and things like that. Our actual assembly here is this FASTA file, contigs.fa. Okay, so if we were to head contigs.fa, we'd see that we've got some this is calling assembled contigs. Now we don't assemble. We're not going to assemble the genome in one long continuous strip because we don't have enough data to do that right now. going to find islands where it was able to string together hashed reads and find overlap. Eventually, paired-in information can help bridge some of those together, as we talked about in lecture. But typically, you're going to have lots of different length contigs that all are different pieces of the genome. So it's still a variety of different puzzle pieces of different sizes. So we'll go back up. So let's see if we can get some more information about our sequences so we can get that insert size and be able to build a better assembly. So this is going to build actually on the mapping that we did. So these things all come back. What we're going to do is we're going to take the reads we used to build the assembly. We're going to take half of them, and we're going to map them onto the assembly as paired-end reads. And then using our SAM file that we get from mapping, we're going to use some of the information, that great information that SAM is good at providing, to look at the column that tells you the distance between mate pairs. And we're going to average the distance across all of our mate pairs that map. And that average should be pretty close to the insert size that was instituted during library prep. So we can kind of work backwards bioinformatically to figure out de novo what the insert size was. And so to do this, we need to use BWA. You could use Bowtie if you wanted. And we're going to be using the contigs file as our reference. And so we need to index it. So we'll go ahead and index that. And then I don't want to map all the reads because it'll take a little bit longer. So let's just pull off half of the reads. And again, you could use seqkit sample to take half the reads. Or we could use head if we want to do it really simple and say, rather than heading the first 10 lines, let's head the first 400,000 lines. That is 4 million. There we go. 400,000. And we'll head our F1 FQ, and we'll just call this temp paired end 1 dot fastq. Okay, and then we'll do the same thing for 2. And now we've got some reads that are not all of them, so it won't take forever, but it's enough to get us the information we want. So now we're going to use BWA mem like we've used before, and we're going to use it on that contigs reference that we already indexed. And we will map our temporary paired end 1 and temporary paired end 2, and we'll call the output a very ambiguous temp.sam. Okay, now that the mapping is done, we could go ahead and tail temp.sam just to see what we got. And we can see, yes, this is a SAM file. It's got lots of good information for us. And now let's go ahead and work with this kind of in a clever way. And these are things that you've kind of come close to doing already, but we'll bring some things together to get some information. Again, remember, we want, basically, if you were to look at the SAM format, we want the 9th column. The 9th column in the SAM format houses the distance between reads where they mapped relative to their other pair. And so what we want to do is we only want to calculate this for reads that actually mapped. We don't want to screw up our averages by including things that didn't map or mapped as chimerics, things like that. So you can go and look at what these flags are, but here we're saying the big F, remember, is an inverse. So basically it's saying don't include anything unmapped. You could look at what the 3 flag and the 16 flag also stand for and try and figure this out for yourself. So we're going to cat our SAM file. We're going to filter it based on these flag conditions. And then we're going to use data mash, which is just a tool for calculating on columns, and ask for the mean of column 9. And so you could build these up one by one if you want, which is something I always recommend. So cat temp.sam. Okay, that worked. We'll hit Control C to stop it. So then we'll pipe that to SAM tools view -f for little f 3 big F 16. Okay, so now we're just getting fewer reads because we filtered it. And then we will run this through data mash, and we want the mean of column 9. Okay, so our estimated insert size was about 160 base pairs. Now, going back to the original paper, it was actually 180 base pairs, so pretty darn close. Okay, so we got within the ballpark, which is good. So now we can actually use this information when we redo our assembly. So what we're going to do is we're going to basically build a new hash map, and then we're going to assemble those hashed sequences, but provide more information this time. So here we're going to do the velvet H again. This time we're going to call it the output velvet PE to signify that it's a paired end. And we're also telling it that these sequences are short paired data. Okay, so we're hashing this out. And when it's done, we're going to go ahead and assemble them using velvet G on the velvet paired end that we just built. Now here we're going to tell it to actually what the insert size is, so the insert length here. And again, if you want to read about more options, you can look at the help or the manual for velvet. And so we're going to say, even though we know the actual insert size is 180, let's just say that we didn't know that. And so we're going to go with the 160. And we'll also say that the standard deviation, and you could have calculated that also with data mash if you wanted to calculate the standard deviation, is 20 base pairs. So we'll go ahead and assemble this. So I just want to point out, now that this is finished in a short time, this is not a typical experience if you're doing kind of a de novo eukaryotic genome assembly. A friend of mine from grad school who was building one of the first fern genomes, his assemblies, which he had to do many iterations of, sometimes would take upwards of 3 weeks, sometimes more, and use up to 1 terabyte of RAM at a time. So again, we're working with very small data sets right now. So just keep in mind that these things can scale incredibly, but I'm trying to keep this within the time frame of a given lab for you. So next, what we're going to be doing is we're going to be comparing our short read and our paired end assemblies. Now, to do that, we're going to be looking at metrics like N50 and total base pairs assembled, like we discussed in lecture. Now, we're going to be using a program called Quast to calculate those for us. It's not something we can pull down on Conda. So what I've done is just provided the line for where you can go and git clone it. Now, I just actually started that before I was talking, so it's already finished. But if you execute git clone and the address here, it'll go ahead and download it. If we ls, we'll see that we have this Quast folder here now. Now, we could go through the trouble of adding this to our path like we did for Cowsay, but it gets a little more complicated. And without getting, again, too much into the weeds, Quast actually uses Python. It's a Python language-based program. Now, there are 2 major versions of Python, basically everything 2.7 and prior, and then 3.0 and after, where Python actually changed some of its terminology and formatting. So anything written in Python 2.7 is not compatible with Python 3.0 and later. So Quast, which is written in Python 2.7, can't use the normal Python that we default to using in our servers under the hood that you don't really interact with. So in order to execute it, we have to explicitly tell it Python 2.7 and then where our Quast Python script is located. That's going to be the simplest way to execute this, okay? And so I'm just giving you that as background information why it looks a little more complicated. So we're going to go Python 2.7, Quast folder, and within that Quast directory is the Quast.python or .py. Now, we're just going to pass it our contigs, our assembled contigs files. And you can pass it as many as you want. We only have 2 right now, so we're going to give it both the paired end one, and we will give it the single end one as well, so we can compare them. And we'll call the output velvet single end versus paired end. Okay, and this will run for just a second. It'll always kick out that it has some warnings and things like that. It's actually working. Don't stress too much about it. So now if we ls, we have this velvet single end versus paired end folder. If we ls inside of that, there's actually a number of things. What we want to look at is the report.txt. So we could just cat it. So let's cat the velvet single end versus paired end report.txt. And here we've got finally a set of columns that don't wrap around because they're short that we can look at nicely in the terminal. And so we have our velvet paired end assembly and our single end assembly. And here we've got some nice metrics that I like to look at. The number of contigs that are greater than 0 base pairs, the number that are greater than 1,000, 5,000, 10,000, 25,000, and the number that are greater than 50,000. And so what you want are more of your base pairs held within large contigs, because that means you were able to string more things together. Our single end, we're not able to string anything together into contigs greater than 5,000 base pairs. So, well, our paired end information allowed us to get all the way up to several contigs greater than 50,000 base pairs. So much, much better job. We could also look at what the largest contig was, 3,700 versus 67,000, as well as the total length. When you add all the contigs together, they add up to almost 2.8 million, which is almost the real genome size. So we're only missing a small piece of the genome being assembled with our paired end data, and 1.8 million or so. So missing a good chunk of about a whole megabase worth of information with the unpaired data. We can also see the N50 and all sorts of other good metrics. So this very clearly shows that the input of information regarding insert size with paired end data make a huge difference. How could we maybe potentially clean this up a little better? So one thing we might do is think about cleaning our data first, using clean data to perhaps build a better assembly by having less errors in our actual reads. So let's go ahead and run fastqc on one of our sequence runs. So we could fastqc data f1.fq and go ahead and fastqc that, and then open it up after downloading it with CyberDuck or your Xterm file manager. Okay, so here we have the first fastqc result. We've got 800,000 reads. Ooh, and these look not great. You know, these could probably benefit from some trimming. Now, I'm going to tell you from experience that if we try and clean these too stringently, we're going to run into a lot of issues, because if you look at the tail end of these reads, there's really nothing that has base pairs towards the end that are even above a quality score of 20. So we're going to end up having to lop off probably half of these reads and make them much shorter. Now, there's trade-offs here. Do you want higher quality data, or do you want a larger volume of data? There is kind of a balance between those two things. Some information is better than no information. And so I'm definitely not going to do a 30 quality score cutoff for these. Maybe we'll do a 20, and we'll basically start cutting off kind of the tail of a lot of these where it dips below 20. Just to show you that we can use different tools for the same job rather than Trim-O-Matic. You could use Trim-O-Matic if you want, but let's try and look at Sickle. Sickle is another trimmer, and so we're going to use Sickle in its paired end application to trim both F1. We could look at the quality of F2 as well, but let's just assume they're somewhat similar for the sake of simplicity. And so here it's asking for -F and -R. That's the equivalent of asking for the forward and reverse reads in the pairs. So _1 and _2 is kind of what that, how we've been referring to those things. We'll call the out data, we'll call the first one F1 clean. So these are going to be the forward reads that come out first. And then we will have our secondary output, which will be -P. This will be our F2 clean. I don't know why I put an _2 there, but we'll just do that. Fast Q. And then all of our singletons. So the ones that lost their other pair, we'll call F clean singles.fastq. And to show you the end of this, we're going to do a quality score cutoff of 20, and we're letting it know that it is a Sanger type of sequence reference. Okay, so now that that's run, we get a little bit of breakdown. We can see how many paired records were kept. We kept around 520,000 pairs out of the 800,000 that we started with. So we already, even using a 20 quality score drop, we discarded quite a bit. So let's go ahead and use that to try and build a better assembly. Now, to kind of expedite this, I'm going to start copying and pasting a little bit. We're going to now kind of rerun our assembly. And you'll notice my names are a little different from below because I mistyped the output name for one of the things. It actually doesn't matter. I'll just go ahead and adjust as I'm typing these out. And so we will hash these now cleaned reads, and then we will build a genome assembly from them. Okay, so next I ask you to kind of repurpose the Quast script I already showed you to now compare the clean versus uncleaned assemblies. And so there's a couple of different ways you could do this. You know, if you're not all that comfortable with looking at this, I would just go ahead and put this in here, and I would start to edit it. So we want to compare our original paired end, which are already up there, against our clean paired end. So velvet clean PE contigs.fa, and then we'll call this clean versus paired end, just for simplicity. Okay, so let us go ahead and look at the output of this. So if we ls, we've got our velvet clean versus PE. So we'll cat velvet clean versus PE report.txt, and we'll see. Now to me, this was initially a little surprising. Our clean one did worse. It did not assemble anything over 10,000 base pairs as far as contig size. In fact, it actually, I think, may have done worse than, it may have even done worse than the single end treatment. So what happened here is we ended up throwing out too much information in our quest for perfect quality data. So not only do assemblies sometimes take a long time, particularly when you're dealing with large genomes, you also have to do many, many iterations of genomes, different k-mer sizes, different quality thresholds, all sorts of different things to try and tweak that balance of information versus quality of information until you really can get the best genome that you can. And there's other ways of using different assemblers and then also conjoining information from different assemblers as well. It's a whole field in and of itself, to be honest. So finally, what I ask you to do towards the end is to vary the k-mer size on your own. And so all you're going to do, I'm not going to go through all this one, but you're going to go back to using Velvet H and Velvet G, but rather than 31 for your k-mer, you're going to use the paired end data and pick a different k-mer number. It has to be odd, and it has to be less than 192. I think the first time I did it, I chose 51. A k equals 31 is pretty standard, and so you may or may not find a k that actually does better than k equals 31, but pick one, and let's see if someone does find one that outperforms k31 for the unclean paired end data, go ahead and share it in Slack. It'll be cool to see if we find a parameter that's more efficient. The final thing I want you to do is to annotate your best assembly. So maybe it was the uncleaned k equals 31. Maybe it was the k that you did as your own optional one. But what we're going to do is we're going to install something called Prokka. It's a little bit of a complicated install, it looks like. This is because there's some incompatibilities between different BLAST versions, and so we had to kind of trick it into running a different version of tBLAST than it wants to default do. And so sometimes figuring out how to get programs to run with little funny workarounds is a big part of bioinformatics. And so I kind of already have gone to the trouble of doing that for you so you can execute all of these lines and get Prokka up and running. So while this is finishing installing, what we did is we made a new environment, which typically if you look at your command prompt, you'll see that it says base over here. It's the basic conda environment. We're going to make some changes, and within those changes, we are going to house them within a new environment so we don't mess with our old existing base environment. And so we're going to call this environment Prokka_env. And so when we actually go to invoke and use Prokka, we can't just use it when we're in our base environment because we've tweaked it to actually be able to work in its own environment. So anytime we want to use Prokka moving forward, we have to do conda activate the Prokka environment, do whatever we want with Prokka, and then do conda deactivate. Okay? If that doesn't make sense to you, that's okay. Just always think of this as being part of how to use Prokka, and you'll be okay. Okay, so let's go ahead and do our run of Prokka, which is, again, going to annotate our genome. What this means is it's going to use a variety of different blasts and searches against known annotated references to see, okay, here's a region of similarity to an E. coli reference genome that we predict it has the same function. And so we're going to annotate this gene as some kind of lactase gene, something like that, from this region to this region. And it's going to do this in lots and lots of different ways. And so what our output will be a .gff file, which is basically that genome with the annotations associated with it. So to run this, we're going to go conda activate Prokka environment, and see we've got our Prokka environment up. And we'll go Prokka. We'll give it our output directory that we want. I'm going to call this K31 paired end Prokka. And the prefix, basically, what do we want all of our sequences and genes and stuff in our genome to be named? So I'm just going to name it after the species that these are, which are S. aureus. So I'm going to call it Soreus. And then we need to pass along what genome we want to annotate. So your folder name will vary from mine, depending on what you ended up you wanting to use as your best one. I'm going to use my Velvet paired end. So Velvet paired end, and we want that contigs file. And we'll hit enter and let it run. Okay, so once this is finished running, we're going to want to take a second and actually try to visualize something. So if you look at your output from Prokka, so we'll CD into what I called mine K31 Prokka and LS, there's a lot of stuff in here. What we want is to download to our computer this Soreus.gff. With this, we can go ahead and look at it in a genome browser. In this case, we're going to use Artemis, which is optimized for looking at bacterial genomes. And so I've got directions for here for how to go ahead and download Artemis. It's a graphical piece of software, so you need to use it on your local computer. And you may need to download a version of Java 11 specific to your operating system. And then you can go ahead and open the GFF file within there. Okay, so once you open Artemis, it's going to ask you to set a working directory. And so I just set it to the folder that I downloaded that had my Prokka stuff in it. And so you hit OK. And then you go to File and open a file manager. And we go into our folder and we select the Soreus.gff. And it might kick back a warning or something. That's okay. And so now we've got basically a browser of our genome. And so within this, we can scroll around and look at different things. We also have a list of all of our genes that were annotated. We can right-click here and say Show Products. So we can actually see what these things are. Here's an ammonium transporter. We could actually click on this and actually go to and see where it's actually located. We could just kind of bounce around. You can customize whether you want to have all this extra information. Here it's showing the sequence and then both the 3 possible translated reading frames from either strand. So that might be more information than you really want. You could also set this to show all sorts of different things. So you could have it show start codons, where start codons are located in different reading frames and all sorts of fun stuff. So you made this. You took a bunch of 101 base pair sequences. You strung 800,000 of them together times 2. And then you went through the whole thing and found all the genes that match genes from known bacterial references and annotated them. So here's the enterotoxin gene, where it's located. It's just downstream of this KJCBL gene, something like that. So this is all, now we're getting to, we could sequence more of this bacteria and map it to this reference we just made and see which genes house SNPs if we were working in a non-model. So now we have something that's actually a nice, important piece of information that can enable all sorts of experiments from this point forward. So hopefully you thought that was a little cool. I think it's kind of cool. And I'll see you guys later.